Oh, thank you for your introduction. It's a great honor for me to chair the first session. My name is Chunyu Li. I'm working in Guangdong Academy of Agricultural Sciences. For the banana researchers, scientists, we all know the Fusarium wilt, black silk toca. However, there are another kind of the banana disease, the bacterial disease, also threatening the world banana development. And for Asia, Africa, different environmental conditions and crop variety structures result in different bacterial disease. In the first section, we invited two speakers. One is from Australia, the other is from Malaysia. Let's begin the first presentation. The speaker is Dr. Nandita Pathania. She is a senior plant pathologist with Queensland Department of Agriculture and Fisheries, Australia. She has more than 13 years of experience in bacterial disease diagnostic research and extension of tropical plant disease management. The title of his presentation is Status of Bacterial Disease and Their Impact on Australia Banana Industry. Let's begin. Thank you. Hello, Nandita. Your sound was muted. Please.
Yes. Good afternoon, everyone, and thank you so much for the introduction and giving me an opportunity to talk in this webinar. So today's topic for this presentation is, would you open my presentation or I have to share?
Okay, Dorothy.
Yes, we will play.
Yeah.
We will play it. Chris,
Dorothy.
Yes, we will play it.
Okay.
Just give us 1 second.
Yeah. So today's topic of presentation is the status of bacterial diseases, their impact on Australian banana industry. So I would like to give a brief overview of Australian banana industry. It is the largest horticulture industry in Australia and is the #1 selling fruit in supermarkets. It contributes around $600 million to the national economy, and major growing areas in Australia are confined to the coastal areas, and mainly is the tropics of Queensland, which constitutes about 94% of production of bananas, and rest of the is grown in the New South Wales, Western Australia, and in the Northern Territory. The major commercial variety is Cavendish, which accounts 97% of banana production, and 3% is produced the other varieties like ladyfingers, GDAAS, and plantain. There are around 800 banana growers in Australia and 130,000 hectare land under banana cultivations that produce 381,000 tons of banana every year. And I reckon that equates one-day consumption of the major banana production countries like India. The biosecurity is the major concern of the Australian banana industry, and pest and disease management are the key priority areas of research and development. Regarding banana bacterial diseases, the corn rot and mochilo disease are the major concern among banana growers. Corn rot is an endemic disease to Australia, and it affects all crop stages of the banana crops. In plantations which are raised through bits and the suckers, the major incidence of disease has been seen on first and older ratoons, which contributes about 36% losses to the banana production. Whereas in case of tissue culture raised plants, the plant losses are mainly in first return, and it causes 20 to 40% losses to the banana plants. Generally, the corm rot has been observed in the month of January to May, when the environmental conditions are quite warm and humid, and that are very congenial for the development of the disease and increasing the load of the inoculum. And second is the mocular disease. It is a minor disease in Australia and occasionally occur on 1 to 2 fingers on the hand, but seen throughout the year in certain parts of the banana cultivation. The external symptoms of the bacterial corm rot is often confused with the fusarium wilt infected plants. They show the yellowing and browning of the lower leaves, but when they are cut open at the ground level, you can see the black discoloration and rotting within the corm and the roots, and it is quite evident in the pictures. And even the highly infected bacterial corm rot infected plants able to produce the bunches, but the plants do not support the weight of the bunch, and often the infected plants tip over at the ground level. For the establishment of the bacterial crown disease, the preferred conditions are warm and the humid conditions and the waterlogged conditions, and generally the poor drainage and occurs more in the poor drainage area of the plantations. The transmission is often introduced through the asymptomatic planting material, especially in a newly opened plantations, and then it is spread through the contaminated equipment and machinery doing the interculture operations like desuckering and the de-leafing, and it can survive long in the soil surface, surface water, and in the plant debris. And nematodes and banana weevil borer damage make it more vulnerable to the bacterial crown rot. And this pathogen has a wide host range, so they can survive longer in the fields. Regarding the diagnostic of this disease, historically in Australia, it has been diagnosed on the basis of morphology, physiological, and biochemical tests using the biolog microidentification system. And the new approach of the new molecular approach has changed the taxonomy of number of pathogens, including the bacterial crown rot. Historically, the Erwinia keratovora, Erwinia atroseptica, and Erwinia chrysanthemi was identified as a major cause of bacterial corm rot. But now, with these molecular approaches, has changed the taxonomy of Erwinia chrysanthemi, which is now Dickia chrysanthemi. And Dickia chrysanthemi is now broken down into different genres and species. And that's why we don't know exactly now what is the cause of the major cause of bacterial corm rot in Australia. Next slide, please. So at the moment, we are doing the depth study of all bacterial corm rot isolates currently and the historical isolates. And in this process, we have recovered 2 more species, which is Fanzong Dai and Zia. And still we are looking at the different genes levels to confirm that whether they are already present and was earlier identified as one of the strain of Dickia chrysanthemi, or they are new to the banana production in Australia. So these are some of the learnings from the past projects regarding the disease management strategies, and it is quite obvious that the integration of all of these disease management strategies reduce the incidence and spread of bacterial corm rot, like use of disease-free planting material, that is the tissue culture plants, avoiding the water stress during the warm and dry conditions, disinfect desuckering tools, by improving drainage of the waterlogged areas of the plantations, improve soil health using the decomposed organic matter or the ground cover, remove the infected plant material, manage the weevil borer and nematode damage, and apply preventative sprays. And it is always good that the come clean and go clean is always work for all of the diseases, which has been seen to restrict the movement of fusarium wilt, which was the recently a major threat to the banana production in Australia. Symptoms of the mochilo. Mochilo is just termed as because the symptoms looks like the moco disease. That's why it is called as the mochilo or the fingertip rot. The infected fruit from this disease has the bulbous base with the thinner tip, and the fingers are often straight and they align out of the bunch, and the internal fruit shows the discoloration, the reddish discoloration, and often confined 1 to 2 fingers per hand. So I'll just go to the previous slide. So number of the different bacteria has been recovered from these symptomatic fingers like Costoconia coani, which was formerly named as the Enterobacter coani, Pentoiglomerans, formerly known as Erwinia herbicola, and the Pseudomonas tollasii and Dickia species and Glabacella species has been found associated with these kind of symptomatic fruits. Next slide, please. So out of these bacteria, 2 bacteria, that is Pentoiglomerans and Costoconia coani, was predominantly recovered from these symptomatic fingers. And we tried to prove these false postulates. And you can see here that the artificial inoculation in the field of the green fingers, you can see that they have showed the straight fingers and they are aligned out of the other fingers in a bunch and produce the similar symptoms internally what we received the infected samples from the pack house, and which prove the postulates, and which shows us that there is not the one organism which can cause this kind of problem, but there are a number of the bacterial organisms which may cause the fingertip rot under certain environmental conditions. At the moment, we are not having a very clear indication of the disease cycle of this pathogen. Likely naturally occurring bacteria on flowers or in the environment may be the cause and transferred by the pollinated insects like bees and ants. And most of the times we have seen the ants nesting in these infected fingers. And the major gaps in attaining this knowledge are the external symptoms are not always obvious, and we receive the sample size that is low, 1 or 2 bunches over the year and 1 or 2 infected fingers, and mostly they are identified at the pack shed or in the supply chain or by the consumers, and which are not always traceable back to the field. And the main is that the different organism associated with the symptoms which needs the further studies to confirm the major cause or the epidemiology of this disease to better devise some management strategies. At the moment, removal of the abnormal shaped fingers at the pack house shed and improving the insect pest management may reduce this quality trait, and which can affect the consumer, has a direct impact on the consumers. At present, Australia is quite fortunate that they are free from the devastating impacts of these exotic bacterial diseases like moco, Xanthomonas wilt, and the blood disease, and heavily rely on the biosecurity and quarantine. And Australia has a great strategy for the regional, national, and international to confine the impact of these bacterial diseases. And along with the phytoplasmas, now it has been recently identified from the PNG, which is at the doorstep of Australia, and it is a great threat to the banana industry. Next slide, please. So future research approach of the banana industry is to increase the awareness of endemic, exotic, and emerging pathogens through using through a survey and increasing the survey and surveillance and extensive molecular screening, and increase the access and validate diagnostic methods to identify the genetic diversity among the pathogenic strains, increase studies on disease epidemiology to devise better disease management strategies, and to increase collaborations to increase the diagnostic capacity and capability for the sustainable banana production globally. Next slide, please. So these are some of the references of the past projects which were done on the bacterial comrade, and some of the other valuable resources which were used to make this presentation. And with this, I would like to acknowledge the Queensland Government Department of Agriculture and Fisheries, Hot Innovation and Banana Fund to conduct these studies. Next slide, please. And lastly, I will end my presentation with this beautiful banana plantation smart farm, which is 100-hectare private property, and it is looking for the precision farming using the sensors and forecasting and biophysical models for the judicious use of irrigation, fertilization, and the chemicals. And with this, I would like to thank for listening patiently, and would be happy to receive any question further. Thank you very much.
Thank you for your presentation. And anyone who have a question can leave your question ahead before the end of his presentation in a chatting room. You can also put forward your question and discuss with the Dr. Nandita directly. Okay.
Thank you.
Yeah.
Can I ask a question, Dr. Chinyu?
Yes. Yeah. Yes.
Okay, Dr. Nandita, it's a very good presentation. I mean, it sort of gives us an idea of the threat of bacterial disease in Australia. But looking at it overall, I think not much of a question of find only, well, the Cavendish variety, and of course you have Fisheries and everything. But I think bacterial disease on Cavendish is very much it's not as important as Fisheries, but the threat is still there. So I just want to find out from you how serious is the threat? Actually, on the comrade, in bananas, how serious is it on the Cavendish? And yeah.
Yeah, it
Sorry.
wasn't that serious earlier, but the recent years with this increase in wet season and the waterlogged conditions in some of the plantation, it is a serious problem. And the recovery of these two pathogens, I'm not sure it is the climatic change or what is happening, or how it is introduced, or maybe due to some, because it causes a problem in number of other hosts also. So maybe it is introduced in the country with the import of other plant crops. So that's, we are just doing research on that one. And yeah, it is the continuing process at the moment.
Yeah, thank you. The other short question is it's soil-borne, I presume.
Yes.
Yeah. So, in your observations, how long does it remain in the soil? Remain in a very risky state. I mean, high risk, or does it go with the weather? Certain weather is more, certain weather is less. Yeah.
Yeah, I agree that in the certain weather it is more, so it requires the wet weather and the free moisture to cause the disease. If it gets dry, the process goes slow down, and then again when the moisture is more and the temperature is more, it again causes the problem. So it is not persistent throughout the years, it goes up and down.
Thank you very much.
Yeah, I have got some one or two questions. Nandita, shall I ask?
Yes, please.
Yeah. So I'm Dr. Tangivil from NRC for Banana, Tiruchirappalli, India. Thank you for your excellent presentation on these bacterial diseases, particularly comb rot and molecular disease. With regard to comb rot disease, actually this will occur up to 4 months after planting in usual cases. Once if it is hardened or matured, then the incidence will be very less. But in your case, since it is a perennial system of cultivation, how come the incidence is up to 40%, to 25 to 40% in the sucker planted as well as in the tissue culture planted one? One question. The another question is in the management aspect, you have told the preventive spray. So
Yeah.
that I don't know what it is. And the third one is mostly we recommend the cover crop or something like that for providing shadow to the plants so that the attack will be very less. When the shadow is there to the newly planted tissue culture plants, the attack will be very less. So, but in your case, how come I don't know these three things? Cover crop is missing, and another one, the preventive spray, I don't know, and the incidence. Yeah.
Actually, with the incursion of this fusarium wilt in Australia, there is a major recommendation now to grow the tissue culture plants. And the tissue culture plants here are raised through 2 or 3 commercial nurseries. And there is a perception among the growers that the tissue culture plants produce more suckers, and that needs more desuckering, and which expose the plants to number of wounds. And due to that one, there is increase in the incidence. And even the tissue culture plants, they are producing more sucker underneath the plant, and that pulls out the plant when more exposed to the wounds and cuts, and which favors the banana comb rot.
No, but in all cases, the suckers will be there, side suckers will be there. So once when we remove it, definitely wounds will be there. My question is
generally that the desuckering which they do at the height of, I think maybe at the 1 meter or something, at quite high when they desucker it, so they are above the ground level. But in case of the tissue culture plants, they produce all the time and they have to remove it. So we also worked on the tissue culture plants that even in the cutting techniques, when they are producing, leaving more of the callus on the tissue plants, they produce number of suckers. But if they give the clean cut, the production of the sucker is less, and the initial suckering reduced to 50%. So still the tissue culture labs here are quite as a initial stage. They are learning how to do it. That could be the one reason, I guess, they are getting that kind of stuff here, and we have a more problem.
So what about that preventive spray?
Preventive sprays are the initial sprays, like when you are doing in the glass house or something, when there is a damage of the other foliar or other bacterial diseases, which just in the humid weather they come in. So if you do at the nursery stage, it reduces the chances of the soft rot. Yeah.
This is a bacticide, something agromycin, something like that, or some other spray?
Agromycin in Australia, the antibiotics are totally banned. So it is just a combination of the mancozeb on certain copper fungicides. So if they are getting some leaf problem with the fungal and stuff, and they have the damage, even the opportunistic bacteria, they cause the soft rot. So if they apply in the combination, it reduces the fungal infection and reduce the predisposing damage and control it.
But you don't use any consortia of bioagents for all these problems?
No.
No?
No.
Okay. Okay. There's still other 2 questions from the chatting room. Yeah. Just now, maybe you have answered the about the chemical treatment on the comb rot, and Sawan asked the question is there any banana species that resistance to this disease, the bacterial disease? It's the last question we must
Sorry, I could not get your question. Could
Yeah,
you please repeat
you
it?
have some resistant banana varieties to the bacterial disease.
Actually, we
The
have
resistant.
our major variety is a Cavendish. So we mostly produce Cavendish here, and the ladyfinger has less as compared to the Cavendish.
Okay. I also want to discuss with you the Cavendish, peace and walk, and other banana species, the resistance to the bacterial disease. However, we have no time, so later I will discuss with you.
Yeah.